So let me start. So as I was talking to, I forgot your name, Cristiano, right? Cristiano. You're asking me where exactly is Bengaluru? That's my first slide. So this is map of India. I'm somewhere here, southern part of India, whereas frontage is from the West Bengal, which is somewhere here. So we are about 1,500 kilometers apart. So when we think of India, the mean field approximation doesn't work very well. A very structured population, right? And this is my recent picture from my lab group. So the work I'm going to present today belongs to many of these students and students over the years who have worked in the past and who are not in these pictures. And before I start the talk, let me also thank all the funding agencies and who have been extremely crucial for conducting our work. Okay, so let me start. So the focus of the research in our lab is on emergent patterns in ecology, and we take a lot of inspiration from physics to understand these patterns. So we have studied, cellular aggregates with some collaborators who study neural systems. We primarily study in our lab using theory and a lot of experiments, fish schools, emergent patterns of collective motion. We also do field work to study how mammals aggregate, collectively move. In fact, a lot of work that I actually do was inspired by a couple of folks from Netherlands, Martin Scheffer, who many of you might know, as well as Max Rietkirk. During my PhD days, I was really fascinated by reading their papers on the idea of how we can use phase transitions in ecology and how we can use pattern formation and sort of very simple physics-inspired models and techniques to understand these ecological systems. For today's talk, I'm probably mostly going to stay with fish and some general theory beyond group dynamics. Let me start with this really fascinating video where you see a collection of organisms who not only group together, but can synchronously move in spectacular fashions when foraging, when escaping from predators, and many other ecological contexts. And now, what is also very fascinating about these systems is that we can actually use simple mathematical and computational models to sort of produce patterns that look like what we see in nature. For example, these simulations here do very nicely mimic many of the sort of real world sort of patterns that you see when it really attacks organisms, right? So this makes studying collective animal motion a really interesting and fascinating subject from the point of view of both biology of the system as well as the underlying physics and mathematics. Okay. Now, when we start to study, these systems have inspired physicists, mathematicians, computer scientists, and of course, biologists for over 30 years now. Now, when people began to do experiments under laboratory conditions, you invariably have a laboratory like this and a fish tank, where it really becomes hard to study more than a few individuals at a time. And the fact that these numbers are small under laboratory conditions is not merely an artifact. In fact, many species do live in small groups. Although the bigger animal groups, such as huge starling flocks or the kind of fish schools you saw, they are sort of very popular. We see many species do live in small groups and exhibit collective motion in small groups. So there is something very important about understanding these small groups. And we can ask the same set of questions. How do organisms exhibit these emergent collective motions? What are the emergent patterns that are universal across different species? Here I'm actually showing you videos of 5 different species under identical experimental conditions. They're all schooling, or they're all sort of same in some physics sense. We know, of course, biologically, every individual has its own variation. But can we know those? Can we describe universal features? Or are there some interesting differences among them? And that brings me to the sort of the importance of finite size stochasticity. Let me just broadly give an overview before I dig deep into this. Many ecological systems do contain small number of individuals. For example, many species do have flocks, where the numbers are not necessarily hundreds of thousands, but only a few tens to hundreds. And if you think of threatened populations or isolated populations, again, their numbers can be small. And in these intermediate size populations or groups, they are large enough that I can still ignore a whole lot of details of individual variations, but they're not so large enough that I can blindly apply the classic techniques from applied mathematics or the physics, where I can assume that what agents are all homogeneous, and therefore I can use this classic technique. So there is something interesting about this intermediate scale. And what I hope to argue in this talk today is that the noise in finite systems, okay, can take non-trivial forms, and therefore they can also produce non-trivial effects. I'm going to demonstrate this in two contexts. One is the collective motion of animal groups, where you will see theory, simulations, experiments, as well as data-driven analysis of these experiments. And I'll also present a theoretical synthesis of eco-evolutionary dynamics broadly in the context of ecological populations and evolution, how again the noise of finite systems can produce some very counterintuitive results. So that's the broad goal. So I hope you will be convinced at the end of this that you buy my argument. Okay, this is now the more detailed outline. So I have sort of three different parts to my talk. In the first part of my talk, I will present theory and data-driven theory of finite flocks. Okay, and I will then go to evolutionary dynamics of finite flocks, because these are now small flocks. Again, some of the classic way we treat evolution via either adaptive dynamics or game theory, they may not work really well. So we have to account for the stochasticity of the finite systems. And in fact, when we combine the group dynamics with the finite size noise, some interesting results can come out. And finally, I will end with a general theory, a synthesis of eco-evolutionary dynamics in finite populations. Okay. So probably this would take more than half of my talk, and these would be last 25 minutes. And feel free to interrupt, and I'm happy to have discussions as I go along. Okay. So now, to motivate the first part of my talk on how finite size stochasticity affects collective motion. So I'm going to begin with this video of an experiment that we have done in our lab. And when we think of collective motion, the quantity that a lot of physicists and also biologists are interested is this measure of group synchrony, also called group polarization. So this is inspired by the sort of spin models in magnets. So you have a vector for each individual, which represents the direction of motion, and you take a vectorial average to get this quantity. And what I have shown you is basically modulus of this vector as a function of time for this very specific video. And so if the modulus is close to 0, that means that the fish are moving in random direction. On the other hand, if the modulus is close to 1, that means that they're all highly synchronized and moving in the same direction. Okay. So now how have people in the past looked at collective motion? So what are the classic models? So the classic models comes from the Vichek's paper in Physical Regulators in 1995, where and then the follow-up papers by Cousin and others, where they sort of added more realism. And the key assumption is that individuals basically move in an average direction of the neighbors. So for example, if I was now the individual, and let's assume we are all not static, but also moving, then what I would immediately do is, oh, my neighborhood is all moving in this direction, therefore I would now need to turn. But there's some stochasticity that, so this term basically represents the average of the local neighborhood, and there is some stochasticity that you add before you move. And every individual basically iteratively does this on every sort of discretized points in time. And the question that people ask is now, these are all local interactions that every one of us and every one of the individuals is doing. Now, what are the emergent patterns at larger scales? So it's a sort of classic theme. And this is just to show you some simulations. Now, this is an example where slightly more complex model where I not only align, but I also want to be close to my neighbors. If that strength of attraction is dominant, then you have this kind of swarms. They aggregate, but they don't go anywhere. They don't have a synchronized motion, right? On the other hand, if the alignment interactions are more dominant, they come together, but they also begin to synchronously move. If it is somewhere intermediate, some amount of attraction tendency, some amount of alignment tendency, you do both, okay, with some stochasticity. Okay. And also you can use these ideas to simulate really large systems. Right? In this simulation, I think I simulated some 16,000 individuals or so, where each individual was sort of following this very simple local rules of motion. And then if you zoom into any part of it, they begin to form groups, and these groups merge and split. And the different colors in this specific example actually was trying to model migrations. And we find that a small number of individuals can actually help large distance migration. Okay, so this is the classic sort of agent-based modeling approach. Okay, and on the other hand, physicists also developed these coarse-grained hydrodynamical approaches where we are looking at thermodynamic limit, large N, and again you look at the same order parameter. Like I'm switching the term to sort of use the physics term called order parameter, which basically is the same quantity, vectorial average of all the individuals, and then you write down certain dynamics of how this order parameter changes over time. And if you the sort of classic case based on Toner 2 and Ramaswamy and others sort of uses various symmetry arguments to arrive at the minimal model that would have a feature like this. So the first term, so I'm going to introduce some terms now, which I will be using repeatedly. I would call this a drift term, which basically determines the dynamics of how the order parameter is changing over time. This is a spatial coupling terms. Because I'm looking at really large thermodynamic limit, there are these local units of order parameter, how are they coupled to each other? And then, of course, the noise is added, the extrinsic sort of noise term. This is the classic framework that people think about in the large thermodynamic limit. But again, remember, my interest is in the small group size limit. So the moment I go to that limit, I can drop the last term, right? So the question that I'm basically going to ask is, if I have to rewrite this equation for the experimental videos I showed you, what would these two terms be? Is this model correct, or would that be something different? Is the question I'm asking. Okay, so to do that, like good sort of mathematical approach, we first need a theory. What would be the theory of flocking when there are only finite number of individuals? Okay, so basically what I would now like to do is I would like to write down the dynamics of order parameter using a stochastic differential equation where it would have a drift term, as I already explained. It would have a diffusion term. Okay, I would like to know what are these drift and diffusion terms. Would they be basically same as this? This would say it has some cubic form with an additive noise. Is that what I would expect? Or would that be something different? Remember, note that I have dropped out the spatial term because I want to understand 15 to 20 individuals, roughly of that scale. Okay, so let me again give a bit more of background just to be sure that everybody is on board. If we had no noise, we would be in the classic nonlinear dynamical limit, also called the mean field limit, right? A typical physics approach would be to take this limit and add an additive noise, right? Additive noise means that the strength of the noise basically doesn't depend on the state of the variable. It's always set, right? And this is an example now. What if we have such a additive noise? Imagine if there are two stable states, and if you have a large enough additive noise, you would also basically go back and forth between. This is one very simple example of how noise would affect a bistable system. Okay. Now, the question that I'm asking, just to orient ourselves, is that in the case of finite systems, this G of m, the diffusion term, need not be a constant. That means we could actually have multiplicative noise. And the moment you have multiplicative noise, you begin to see many counterintuitive results. So that your expectation based on the drift, where you do analysis of equilibrium, stable equilibrium, and the noise would just make the system sort of hover around this equilibrium, those may not be true anymore because the noise is behaving in a non-trivial way. Okay. So this is the broad framework. And of course, this point has been made by others. So I'm not making this point. Okay. Yes, please.
It's just, I don't think the fact that you get multiplicative noise is necessarily related to finite systems.
Absolutely. No,
you
could get it for many different reasons. Yeah. Yes, absolutely. Right. Yeah. But in for the context that I'm studying, that is one way we can get multiplicative. Okay. And of course, this point has been made by others. The fact that the multiplicative noise can produce counterintuitive results has been made. In fact, this is the book that I studied during my PhD days. That sort of it's called the Noise-Induced Space Transitions by Houston, K. and Lefebvre. And more recently, in the context of finite-sized ecological systems, Alan McCain from which university in UK, I forgot. And many of his students have done really brilliant work. Those are all theoretical works. So where we stand, we mixing, sort of integrating the theoretical framework of those systems with real data of the fish schooling. Okay. So that brings me to the next point in my outline. I will first present now what should be this theory of finite slog. I'm going to use the word mesoscopic primarily to sort of indicate that this is not the thermodynamic scale. This is not the individual scale, but somewhere in intermediate scale where these fluctuations are important. Okay. So now I'm going to introduce the spin model of collective motion. So in this spin model, I'm going to assume extremely simple rules. It's actually simpler than the Vishwesha model that I mentioned in the initial slides. So in the Vishwesha model, just to remind, every individual does an average of the neighborhood and then moves with some noise, right? Here, what the individual do is they don't do an average of everybody. They just pick one of them randomly and then move in the direction. That happens at a rate C. The individuals can also just decide to turn a bit on their own. Yes, please.
So this then is similar to noisy bottom model.
Oh, yes, absolutely it is. It is. Absolutely it is. Great, thanks for the connection. Yeah. Okay. So, so there are two very simple rules here. Now what we basically do is we compute the group polarization for this model. Okay. So how do we do this? So this is a slide. I won't go into details. The basic idea is to define a density function. We then write down the Fokker-Planck equation for the density function of different. There are a lot of individuals, right? So they're all located in various directions. How are they concentrated in the direction space? And then we write down a mass Fokker-Planck equation. And then we sort of use the fact that the number of individuals is not large. But large enough, but not large, to sort of keep only the leading order term in the LSD's expansion. Okay, and then from that we now obtain an equation for the order term. So we start with the angles for everyone, density function for angles, and from that we look at the average of the direction vectors. And then this is how the equation looks like finally. So now let me explain this equation. So what does this equation tell us? So basically, if this is the dominant interaction, C is that rate of turning towards a random individual. What we find is that the rate of change of the order parameter follows this very simple stochastic differential equation. So what does this stochastic differential equation have? So let me highlight two terms here. A basically is that spontaneous turning rate. It's exactly not the S, it's a function of S. And C is basically the copying rate. Okay. So how do we understand this equation? So to understand this equation, I have this graphs. So in the limit of very large noise, we basically can sort of, don't worry too much about this. What we basically have is dm by dt is minus am, which means the order is exponentially decaying towards zero. Okay.
You have mid-large noise, you mean large n?
Oh yeah, large n. So therefore small noise. Sorry. Thank you for the correction. So basically you can think of a ball that is jiggling around close to zero, where m0, as I told you in the beginning, basically represents disorder. The fishes are not really synchronized. On the other, so now if you look at plot the PDF of the corresponding state variable, you will get basically sort of a Gaussian distribution around the order. On the other hand, when n is small, so this term is now non-trivially large. Now let us see what happens. Now this guy is sort of trying to come down because of the deterministic term. But what happens is, as it's coming down, the stochastic term has this feature. It's actually maximum when the ball is close to zero, when the state is close to zero. So because you see that, it's 1 minus m squared is actually maximum at the disorder. So there is actually a tension between the deterministic or the drift term, which is pulling the system towards disorder, and the noise term, which is pushing you away. It's not necessarily pushing you away with a direction, it's pushing you everywhere. And you can actually prove analytically that if the n is less than a threshold value of a threshold value, you actually observe a completely different state. Deterministic stable state is zero. Oops, I shouldn't go too close to it. But the observed state would be actually at +1 and -1, which corresponds to a highly ordered state. Okay, so this is what the theory predicts. Okay, and if you do basically have a small enough group, you would actually find order even if the fish were just copying one neighbor randomly, but you wouldn't find order if there were too many. That's the basic prediction, which is why we call this intrinsic noise-induced order. It's intrinsic noise because the noise is entirely intrinsic to the dynamics of the interaction. It's not something external, right? And that noise basically is pushing you away from the mean field expectation. Okay, and now let me present one more model, which is sort of reminiscent of what the Vichek and cousin models are. Will be a local averaging of the directions, right? So basically, we still keep both of these pairwise interaction and the stochastic turning. We can always reduce this to zero. We get the this model only. Here, there is a new interaction where 3 individuals interact. And when 3 individuals interact at some rate h, which I forgot to write here, for example, in this case, the guy who is most misaligned would align with one of these 2. Or you could also assume they align with the average of the 2. Okay. So, for example, in this case, you can see that this fish has turned towards one of the 2. Okay. So now, if you write down again the same sort of protocol, start with the density function, Fokker-Planck equations, master equation, system state expansion, and you write down the E to SD, you now have a slightly more complex SD. The point to note here is that in contrast, I will now contrast this, I think, in the next slide. This is the slide that contrasts these 2 one by one. Let us look at the drift term for both the models now. For the pairwise interaction model, the drift is basically a linear function, suggesting that the disorder is stable, the red line, whereas the ternary model shows that a value of m close to 1, how close depends on the strength of ternary interaction, actually is now stable, and the disorder is unstable. So just turning away from pairwise to ternary changes the drift function dramatically. Okay, and the diffusion functions are qualitatively similar. They don't really change. Strength, of course, depends on the values of the terminals. So this is the fundamental difference. So basically, both the models admits spooling, highly synchronized motion, but the main difference is that the pairwise copying interaction. Okay, I think I messed up a bit here with the animations. Okay. Sorry. So the difference is that the pairwise copying interaction models admits order only for small flocks, whereas this model admits flocking for both small schools and big schools. And if you want to again think of in terms of potential wealth, which is sort of classic in physics. So here the disordered state is the stable, noise is pushing away. Here the ordered state is stable. Okay. And the noise plays a less significant role. And you basically can have deterministic order in both small and large flock size systems.
Yeah. I really like the picture, like this equation there, but I'm confused on how you ended up with these equations. So it's possible that you can go back to your big slide with the different mesoscopic and.
So did you mean this slide?
Yes. Yeah, the one with the mesoscopic and yeah, this one.
This one?
Because it's sort of.
It's a pretty
like
messy.
It feels like you're going to be going the other way around than I'm used to, because normally you write down, you have your stochastic processes for your individuals.
Yeah.
For these individuals, you write down your master equation.
Yeah, these are the stochastic
Yeah,
processes.
indeed. And then you get your master equation for your stochastic processes. Then
So
you
this
get,
is yeah.
and then you get your Gamer-Moyal expansion or whatever you like, and then you get your Fokker-Planck equation.
Yeah.
But you say you're going
So
the other way
yeah,
around.
okay, well, this is a master. So I mean, I know I made a mistake here. This basically is basically the master equation, right? Because it's basically looking at what the current state is and the influx minus outflux to all of them.
Yeah.
And technically, this is really where I'm using this is when I'm actually converting to a Fokker-Planck equation. I'm sorry.
Okay.
So actually, I think it's a really good point. I think I made this slide quite some time ago and I didn't manage. Yeah, but you're right. Yes. So this is the master equation, right? For the density function phi.
Yeah.
And then I use this Kramer-Moyal expansion accounting for the finite system size. That's when I get the Fokker-Planck, which I have not written down here. And then from there, I sort of invert to the SDE,
Yes.
which in this case happens to be.
But then I'm wondering, so you have these two reactions.
Yeah.
So you have this random de-alignment and you have the align with your neighbors.
Yeah.
So would
So align a neighbor or just a
yeah.
in the first model, align with a random neighbor
Yeah.
or just turn random.
Yeah. But so wouldn't you then expect to have also two deterministic terms in your Ito SDE?
So what happens is that if I copy somebody randomly,
Yes.
okay, and if I copy only one individual randomly, so the terms of influx and outflux just cancels out. But if I copy if I pick two, and then we do that majority sort of rule I mentioned, then the terms don't cancel out. So it's like, C x1, x2. I mean, if I were to discretize this, if you look at the sort of inflow rate would be C x1, x2, where x1 and x2 are two discretized states. Okay. But outflow would also be exactly that, C x1, x2. Okay, somebody else copies me versus me copying. On the other hand, if I do with two individuals, it becomes C x1 x squared minus C x1 square x. That doesn't cancel. Which is precisely why in the ternary interaction model, you do have a nonlinear term, but not in the pairwise interaction.
Yeah.
It's a small difference. Thanks for this. Thanks for the question. Yeah. But you're right. I should fix this in my next presentation. Any other questions? You're most welcome. I think this is where I was. So I think the next slide, where I have sort of, I hope I have convinced that in the pairwise copying alignment model, you will have disorder stable in the Larsen limit, but you can have an order in the small flocks only. Whereas in the ternary or sort of average interaction models, order is stable in both the limits, as long as the strength of interaction are high. Okay, so this is the sort of basic theory. And we also have done sort of somewhat more complex simulations, which has space in it and so on and so forth, and they all align with this analytical expectation, which I don't show here. Okay. Any questions before I go now? Now I'll turn my attention to experiments. Are we good? Sure. Okay. So now what do experiments say? So remember, I showed you this video, right? I showed you this video and asked, oh, look, all of these fish are doing schooling. Are they all the same? Of course, they're not same. But are they all different? Or maybe not. Maybe yes. We don't know, right? The question I want to now ask is, I now have from the mesoscopic theory of flocking, there are two possible ways the schooling can be achieved, right? And they can be achieved by the simple pairwise interaction, but they will be sustainable only in small schools, but also via ternary interactions or the average interactions. But what are these fish schools following? That's the question I'm now asking. So what we do is we do all these experiments and then we track the fish using computer vision techniques. And then by tracking, we can get the direction of each fish. And from that, we calculate the order parameter. So now we have the order parameter time series for the real data. Okay. Here is how it looks like. To plot it for sufficiently long, there are three different species for representative purposes. The gaps you see are entirely because sometimes we cannot track them when they sort of pass each other and for many other reasons. Okay, but yeah, I don't know. They all look sort of what the two things you are seeing are the x component and the y component of the order parameter. They seem to oscillate. The oscillation happens because they sometimes follow boundaries and all. Okay, but they all sort of look somewhat same. If you now plot the PDFs of this order parameter, so what you're seeing here is the two-dimensional PDF of the order parameter. They all look same. You can see the heat map at the edges, meaning that they are mostly very highly aligned most of the time. If you look at the PDF of the magnitude of the order parameter, again, they're basically mostly aligned. There are some differences. It's a bit unclear, because they all have some different parameter values, or is it because is there something fundamentally different about them? At this stage, we just don't know, right? So, but they all have high synchronization is what we can say. So the question now we are interested in is the inverse problem. We have this time series, high resolution time series data. Okay. And when I say high resolution and long time series, every second has 25 frames in the video, right? So, and then each experiment runs for about 1 hour. So every video gives me roughly 1 million data points of the order parameter. And it's a time series data. It's really pretty good, right? Really remarkable. And we replicate experiments. So given that quality data, can I now get equations? Just like I got those differential equations from the two models, right? There I use, of course, the standard physics techniques to get the equations. Can I now get the equations? Because both of them are ordered. Both the models predict that under some conditions, you can be highly sort of, synchronized. So the only way to distinguish them is if I know the equation. Yes, please.
No, just before getting there, why don't you just test your models on the data?
Well, the question is, what would I test against? Would I test against the PDFs? But the PDFs all look sort of
I
same.
don't know, you can simulate your model and you have a time series for that.
And what do I compare with the time series is the question, right? So that's where that's precisely what we do now. So in this, we published a Python package which just came out very recently. The idea of this Python package is that we have a time series data, one-dimensional or two-dimensional. From the time series, we can actually construct the drift and diffusion function. Okay. Okay. So now the basic idea behind the method is that I wish I had another slide where I had these simulations of known models. I forgot to include that here, I think. Okay, in fact, what we did in this paper was also we simulated the SDEs corresponding to the pairwise model, SDEs corresponding to the ternary model. We use the parameters such that they both show nearly identical PDFs. The PDFs are absolutely identical. But could you still distinguish the fact that the drift term was linear in one of them and cubic was cubic in the other? The answer is actually yes. And this package actually implements the basic idea of the Kramer-Mal coefficient. The idea is that if you have a time series, the drift term actually is approximated by the first jump moment of the time series, whereas the diffusion term is the second jump moment of the time series. So this is something.
In the Kramer, they only care about the moment, right? First, second, third. Why you say that you compare visually two PDF? It's impossible to compare.
Of course, we
PDF,
cannot.
right?
Yeah, absolutely, yes. So basic idea is, if you were to only fit the PDFs,
Yeah.
you would not know the underlying equations.
Yeah.
But only looking at the moments,
Yeah.
you can actually distinguish the equations. And that
Can you look at the correlation in time of your moments?
That is
Because I guess
something
it
we
would
do.
difference, right?
we
So-
do look at that. In fact, we use that as a diagnostic tool
Okay,
between real data and that is part of the, I think, somewhere here, yes. So you should not only compare histograms, but also autocorrelation function. But more importantly, we can actually compute infer the drift and diffusion by looking at the first and second jump moments of the type series.
And what do you get?
So what do we get? Okay, so this is the data. Let's remind, this was our data, right? This is for one species. So this is how the drift function looks like as a function of the two vector variables. How do you think this looks like? I mean, the fit sort of indicates that. In fact, you would know very easily, even without the fit, there's actually a plane, basically a linear function. The diffusion, on the other hand, looks like a dome-shaped function. And in fact, a simple quadratic function fits really well. Not only that, the maximum strength at disorder, if you remember the theory, scales as 1/n. And that's precisely what we find when we did experiments with 15, 30, and 60 group sizes. So if you put all this together, this is the equation we get. So the inferred equations from the data has a linear drift term and a quadratic diffusion term. Okay. And the noise is surprisingly uncorrelated. So what we do is, after constructing all this, we compute the residuals between the model and the data from the time series, and this is incredibly uncorrelated. And this was the work of Jitesh, which basically showed that in this species, which is found in southern part of India, the observed synchrony that you are seeing is actually best explained by a noise-induced order, not deterministic order. Okay, and it also sort of gives you some insights on how they are achieving this. So the model just assumed that they are randomly copying a neighbor, which is actually, in my opinion, simpler than averaging neighbors. Of course, you still need the information of, should I pick somebody randomly and then copy, as against moving everybody's direction and then copy. Okay, and this, I think, is a very interesting example of where biologists would expect that the real data is more complex than the simplest physics theory, but what we found with the data was that it's simpler than some of the simplest models that are out there in the literature. Okay,
So is this a general statement for all fish species?
that's the question. So do all fish schools exhibit the same equation, or does everyone have their own equation, right? So that's the next question we have now set out to understand. So I'm now showing you, I think, 5 of them, all of which I think were done in our lab, 5 different species. And we have added a few more since I made this slide. So we have now about roughly 10 species, including a couple of species of experiments which were done in other parts. One with Guy Terrelos in France, and one that was publicly available. So these are tentative conclusions. We have not published this. This is still work in progress. What we find is the following. So if you find the equations for all these different fish species, they fall into basically 3 broad categories. The one is what I already showed you. Here, the swarming or the disorder would have been stable. but you still find a high synchrony. That's now noise-induced order. This is what the classic theory predicted. Classic Wittek-like models predicted the order is deterministically stable. And these two we do find. We have now two species that show this sort of a behavior, one or two that show this sort of a behavior. But in fact, what you find is surprisingly majority of them show this bistable behavior, which actually where the organisms basically switch between a swarming stable state and a schooling stable state. Okay. So basically, we basically find these three classes of the schooling dynamics. Okay. And what is very interesting is that this is something none of the simpler theories as of today, none of the simple models actually predict. So we are currently working on what are the rules that individuals are probably following that produces this bistable dynamics at the group level. So we don't right now have a good theory for this observed class of fish schooling. Yes.
Just a question. So this is double well potential. Why you just make a model with the rhythm derived from a double well potential? Then it answer the question or not?
So can you repeat your question?
I mean, there's a picture, the cartoon show a double well potential, right? And the drip in your underdam Langevin can be also expressed as
so here is,
gradient of that.
I understand your point. So the question is, when I say there are two models, so these are all mesoscopic models.
Yeah.
So these three are basically equations themselves, right? They are basically stochastic differential equations for the order parameter. But the heading of all these are about the individual rules. Pairwise alignment interaction. It's about what the individual is doing, right? And this is the classic averaging, right? So we don't know what are these agent-based or individual-based rules that can produce this bistable at the group level. Is that clear? Is that why I have to...
we work on that kind of problem.
Yeah. So basically, we know the group level behavior now, group level SD, but I don't know the individual level interaction. By the way, student has worked out, but I haven't put because we have to still discuss and make sure it's, it's all rigorous some kind. But which is why I wrote cautionary preliminary findings.
Because in the opinion dynamic literature, there's a rule at the agent level, we produce that double well at the group level.
Yeah. I would love to hear about those work. In fact, we are actually currently also looking at the opinion dynamics literature, how whether we can borrow some ideas from there. One thing we find is that the actual motion, the speed seems to matter a lot, for which I don't know if there's an analog in the opinion dynamics literature. So what you find, which I have not shown hidden slides somewhere, are that so far I have focused about the group synchrony. Okay, now what you find is surprisingly that there's an interesting coupling between the group synchrony and the speed of individuals. So when they are moving fast, they seem to move in a highly coordinated fashion. The moment they slow down, they also become disordered. So this is something I don't think there is an analog in the opinion dynamics, but maybe I'm wrong. Maybe there's some order parameter transformation one can think of. And we now are working on a sort of a model that sort of produces these observed features. Yeah. We have a couple of candidate models. We have to find out which is now more realistic and which is also more parsimonious. Some combination of realism and parsimonious. Parsimony, yes.
I think can I just ask, is there a sort of evolutionary explanation for why some species just look linear and others are Toner 2-like?
Yeah, absolute beautiful question. I don't know the answer. We are trying to understand.
The ones that do linear, just fish tank fishes, or are they
I don't know.
in the wild? Or?
I don't know. That's a great question. But I have. In fact, we thought maybe I will tell you how we tried to address this.
Yeah, you could ask, is there a strategy for better flocking?
Exactly. Yeah. So that depends on the ecology, right? So is it for foraging or is it for predation?
Right.
So what we have tried to do is run the simulations with these two. We haven't done this. These two models where individuals follow the pairwise alignment versus the averaging. And then if a predator attacks. For example, the success rate of a predation doesn't depend on which rule do they follow. So it's a bit hard to disentangle because for every success rate that I can achieve in one model, I can achieve in other model using a different parameter. So now, yeah, so I don't know right now, but there must be some reason. There has to be some reason.
Or maybe it doesn't matter.
Maybe it doesn't matter. I don't know.
Because the behavior that you care, both of them do the similar thing with different parameters,
That's-
like speed and stuff.
Yeah, possible. Yes, yes. I don't know. Yeah, we need to understand this.
Yeah, I was thinking that maybe some of you remember better, but a few weeks ago, you had this guy talking about sheep flocks who also
Yeah,
just.
Pelandor.
Sorry?
Pelandor.
Yeah, it was just we also said they just had this grazing behavior, and then suddenly at some point they decided to
Yeah,
walk in a certain direction and then start grazing. How does it transition from you're all grazing to all moving in one direction to a new plot? How does this work? And I think this is probably something the same thing you see in here, this transition between
Yeah.
disordered and ordered versus
Yeah.
transforming and schooling. Yeah.
But let me just say something very interesting here, though. In all the three cases, the histograms of the order parameter is highly skewed towards high order. You know, there's a thing, if you look at the histogram, I wouldn't predict that there was a swarming stable state. Okay. So that's something I would just, there would be no way for me to know from the histogram. So there's something very subtle here. The interplay of noise and the underlying tendency dynamics is very interesting. Yes.
Instead, that fact that the histogram is so focused towards alignment, not an effect of the small tank. But they're always on the
That's a
board.
possible scenario, but we have done with species, other species. Not every fish species would align just because they're in tanks.
Okay,
We have discovered about 10 different species because they don't show high schooling. Now you ask me why did we not study them? From a purely methodological point of view, it turns out that the moment you're in a swarming state, the underlying SDEs become degenerate. They all become same. Okay. So it doesn't reveal much. That's the main reason why we don't study disordered schooling behavior. It doesn't really reveal much in terms of the methods vary. There might be other methods.
And maybe it's also maybe a matter of definition, because you have this histogram, this order parameter going from 0 to 1. You say, okay, well, around 1 is highly organized. So maybe if you just call everything from 0 to 0.8, if you call this swarming and then just group it together in one big block, then you probably would get the histogram with a very clear swarming and a very clear schooling. So maybe it's just
Why
that,
would I have been like that?
well, it's just that maybe if you have a school that is swarming, at some point they will just magically all move at some point in the same direction or not, because you have all these random angles, they won't be always 0 exactly. So maybe it's just that your bin for swarming should be somewhere anyway from 0 to 0.5 or 6. That's just me now guessing from the top of my head, but just thinking about what's the bin that you call swarming, you would just
So here I am not, you know, to an extent possible, I have not used any bin measure for swarming. I am assuming that 0, I'm clearly saying 0 is swarming and 1 is swarming.
Yeah.
And I mean, I don't know, if you look at the histograms here, you see that it's really close to 1. And it actually is 1. The peak is actually 1. In fact, if you do a kernel density approximation, if you plot the kernel density, if you actually overshoot 1, it's so highly peak towards 1. I mean, I sort of get your broad point, but I would argue that it doesn't quite apply to this. If it was a bit ambiguous, imagine if it was a flattish curve and so on and so forth. Some ambiguity of how I've been might arise, but I think it's sort of crystal clear.
does the sharpness of the peak scale with the size of the individuals, because they're effectively in a smaller tank?
It's a great, I haven't studied this, but we do know that in general, the juveniles of many species do schooling. And whereas as they become adults, many species do lose schooling. They sort of become pairwise male-female pairs from each. Whereas, so all of our studies are with juvenile of roughly same size of around 2.5 centimeters. Some variations will be there because we depend on what's available in the market to buy fish. But by and large, we know it also makes sense because juveniles are highly prone to predation, which is also why and it's a good strategy to be grouping and schooling when there's predation. Okay, so so basically, sort of summarize this part of my talk. So this was the experiment with finite plots, where the numbers are actually small. It introduced a stochastic effect, which is unique to the finite sizeness, which we were able to sort of decipher using this data-driven. It's called data-driven equation discovery. I get the data and then I discover the underlying differential equations, right? That basically suggests us 3 classes of collective motion with a bistable twist that doesn't exist right now in the literature. Okay, so now I just want to add a note of caution. If there are people in the audience who have worked on this field for a long time, they often ask me this question. Of course, fish do a lot more complex things. Every organism does many more complex things. Therefore, it's actually, I would argue that to know exactly what an organism does to remain in a spool and be highly synchronized is actually an incredibly hard question. What we are trying to do using this modeling framework is effectively, in terms of alignment, what are they doing? Okay, and it doesn't tell us how the alignment itself actually came about. For example, in the literature, people argue that attraction can also cause alignment. You don't need to do something called explicit alignment. This modeling framework cannot distinguish those detailed features because it's not meant to. We are really looking at the group level mesoscopic equations, right? And I also would like to argue that what do really animals do, which is a question that a lot of people want to understand in this field, depends on what are we trying to explain. Do I want to explain how far the fish are from the boundary? Or do I want to explain how synchronized they are? Do I want to explain the correlation patterns? Depending on what exactly I'm trying to explain, my conclusions about what fish are actually doing will differ. Okay. So in some sense, without the context, the question of what exactly animals do, in my opinion, is actually not even a well-posed question. Okay. Yes, Sam, you had a question?
Yeah. Maybe it's off-topic question, but like you use underlying overdam Langevin. What happens if you take into account the speed and then consider underdam Langevin? Like is this Python bucket? You also have this data-driven discover underdam Langevin?
It can, yes. It can. In fact, in the paper that I present, I sort of briefly referred to, we have a cell migration data, okay, where people had previously argued that basically it's a cell that goes between 2 compartments. If you look at time series, it looks like a oscillatory dynamics. Now you can model oscillatory dynamics in 2 ways, right? One would be that it's like a van der Pol oscillator, right? Other would be that it's if you look at the experimental setup, it's basically there are 2 wells. Cells sort of somehow moves back and forth, which is almost analogous to saying it's a double well potential. Okay, what we show is that the method can distinguish the the underdamped system, like the way you are saying, or an overdamped system. It picks only one of them, not the other. In fact, it's more of a close to a van der Pol oscillator than a double well potential oscillation. So it actually is positive. Okay. So now I don't know how much time do we really have.
Yeah, you can go on, I think.
What's the time now?
It's like 5 minutes to 12.
Okay. So I mean, just tell me how farther I want to. Entirely depends on. So now now we have seen how finite size noise affects the within lifetime group dynamics of individuals. I have also been very interested in understanding my evolutionary dynamics. Okay. So I'll show you how finite size noise also affects evolutionary dynamics. So to do so, I will use this classic example of our dilemma of cooperation, where there are 2 actions that each individual can do, either to do a cooperation or a defection. Okay. And it's sort of very, very well known that cooperation is unstable to invasion by defectors or cheaters who don't do anything. So cooperation is an action that benefits others. It gives me a cost. Why should I do this? Makes no sense from an evolutionary Darwinian perspective, right? And this is how the classic payoff matrix for the cooperation looks like. Now, this shows that, if there are two cooperators interacting and they both get B minus C payoff, but a switch to defection always increases the payoff from B minus C to B. So therefore, I should always be a defector, independent of what others are doing, right? So then it has been a question that people have pondered and thought about for a very, very long time. And there are many mechanisms you overcome by which you overcome defection. And one such mechanism is the assortment via spatial separation. For example, here you see that there are cluster of cells growing. Okay, if you sort of label one of them as a cooperator, other as a defector, what you can see here is that if an individual interacts only locally, right, the blue guys will interact only with blue, and the red would interact with only red, except at the edges, right? So you can also see the edges are sort of separating over time. They're not interacting as much. If that happens, cooperators will be interacting only among themselves, whereas defectors would be only among themselves. And this would now avoid the defection by defectors. So broadly, the general principle is that if you can somehow achieve assortment, which is probability of cooperator interacting with the cooperator, minus the probability of a defector interacting with the cooperator. If the difference is more than C by B, the cost of cooperation by the benefit of cooperation, we can achieve cooperation. Okay, and this is called Hamilton's rule in a very general sense. There are a lot of debate about how to interpret these terms. I will not get there. Okay, but it's possible. So the idea is somehow cooperators must make sure they interact among themselves and somehow avoid interacting. That's the basic idea. And this provides a mechanism. And in this context, it's generally thought of mobility would destroy cooperation, right? Because if you make all these particles now move a lot, not just be locally where they are, how would you maintain this segregation? If you don't maintain this segregation, there's no assortment, therefore there's no cooperation. So this is the sort of broad thinking. But what we showed in one of the papers is that if you now introduce the prospect of grouping via the same mechanism of this alignment and attraction, you can actually achieve cooperation. So I will just show you how, and I'll show you sort of over the next few slides how finiteness of the system actually becomes very important. Okay, so I'll show you some simulations to sort of make the story mostly pictorial and visual. Okay, this is an example of there was a parameter R0 on the beginning. So it basically tells if R0 is 0, I'm basically not interacting with anybody. I'm a random walker. This R0 is very large. I talk to a lot of guys around me, and then I move in the average direction of all of them. If it's only small, it's only local neighborhood. So RS basically decides the extent of neighborhood of interaction. Okay. Happened. Here you're seeing for RS is 0, which means you don't interact with anyone. Basically, everybody is a random walker. If RS is somewhat intermediate values, you find cluster formation, but not big clusters. They're all small clusters, and small clusters are very important. If RS is very large, you form massive big groups. Okay, so this is something we know from collective motion literature. Okay, now that basic idea we introduced here was, in addition to this RS, individuals can also have a tendency for cooperation. Okay, think of it as just literally discrete traits. I could either be a cooperator or a defector, while I am also moving in all these groups. The question is, will cooperation sustain in this sort of very mobile population? Okay, so now let me show you again a few videos. Let me just know by looking at the videos, do you think there is some clustering of two colors here? So here, what I have plotted, and what I'm showing is a scenario where the cooperators and defectors, they have equal range of interaction. If we do this, so red, I think, is cooperator, defector, and the blue is cooperator. Now, if I pause this at any random point, okay, sorry about this. You can see that any sort of group has equal likelihood of having cooperator or defector, right?
So is this
There might be-
pairwise interactions or?
Here, I'm assuming a local, that RS is the radius of interaction. Within that, I interact with everybody.
Okay,
They're more of classic models. Okay. So if that RS trait is same for everybody, then the cooperators, defectors are sort of well mixed.
so
Which is really-
you're running the social dilemma game?
Yeah. Now imagine if they have a different, what I now showed you was a scenario where they have different interaction ranges. The blue guys are all now separated out. Red ones are all separated out. So this is another simulation with a fluid, hydrodynamical model, which is not too important. So let me just pause here and see that in this cluster, there are mostly blue individuals who are cooperators. There are many other clusters which are all defectors. So what this shows you is that if the cooperators can somehow interact more strongly with their neighbors than defectors, cooperators can form groups which are mostly their own type, even when they are moving. That's the basic idea. So in fact, what we did was we now did simulations where we let each individual evolve the value of how much they interact and whether they can be cooperators or defectors. So they basically the co-evolution between cooperation and grouping behavior. And
You're not allowing for strategy change.
we do. Cooperators
But
can become defectors.
Before you were not.
In that, I was just showing you a representative video
Okay.
within the lifetime. But at the end of lifetime, everybody now measures their payoff,
Okay,
which is based on the prisoner's dilemma, payoff matrix, but generalized to a group size n.
so you're grouping C's and D's to.
Yeah. So in the prisoner's dilemma, you have pairwise, only two individuals, right?
Yes.
We now sort of generalize that payoff matrix for a arbitrary group size G.
Yeah, sure.
And then depending on the number of proportion of cooperators and defectors, everybody gets some payoff, but
Right.
only within the group.
And they share.
They all share, yes,
equally, yeah.
exactly. And every group now gets this, and now again the probability of now switching depends on the sort of classic models of game theory. Okay. So if we do that, what we find is that surprisingly, if you now keep running this evolutionary simulation, surprisingly, we find that there is this assortment sort of spontaneously emerges. So basically, there are actually more number of cooperators than you would expect just by chance. And that happens because cooperators can manage to form these clusters that separate them out from the defectors. Okay, without going into too many details, what we are finding is some sort of a multi-level selection sort of spontaneously emerges. So in the beginning, let me just tell you, in the beginning of the simulations, everybody was a random walker, everybody was a defector. Okay, and then as they are moving, there's sort of there are accidental interactions, and then there is mutations, which corresponds to changes in the strategies and so on. Okay. So we find basically some interesting multi-level selection dynamics where different groups having different compositions sort of effectively compete. And then collective motion and assortment of cooperators and cheaters sort of emerge spontaneously. Okay. And the thing that actually maintains this is the fact that cooperators and defectors evolve to have differential cohesion. They will not have same degree of social interactions. Because they have this differential interactions, they're able to maintain this cohesion. Okay. And not only that, these groups actually are not static. They are constantly moving, merging, splitting. So this sort of shows that even in highly mobile populations, one can actually have cooperation. So now we did a follow-up
Can I
work.
just ask you something? Sorry. What is the passive? What was the difference?
So the difference here is that here there are sort of self-propelled particles.
And the other.
On the medium, there is no medium here.
Okay.
Active particles here, they don't move.
Okay.
unless the medium moves.
Yeah, okay.
So here we were simulating a fluid turbulence in the
Yeah.
background.
And then you put the particles.
Particles now
Okay.
trace.
Yeah.
And then they also have a stickiness parameter. So the stickiness parameter is an evolvable parameter in addition to cooperation and effect. So under sort of very two different sort of movement scenarios, you can actually have more cooperation than. So it is not that the cooperators undergo fixation, to be very clear. So this is not a case where cooperators fully win, but they actually coexist with effectors. And we actually did a follow-up study to understand the role of noise. What we found was that the fact that the groups are small actually was really, really important. And without going into too much detail, so we sort of simplified that each individual can now have only two traits: a tag, which is like a cooperator or a defector, zero. Tag is like the stickiness is the tag, stickiness with other individuals, and then cooperation or defection. So basically, two traits. Each trait has two values, and then we wrote down transition sort of rates for all these different between different strategies. And then what we then did was write down a stochastic replicated equation for this. Sorry, I'm going to jump through this without telling you too much details. It's basically, if we had a classic game theory, you would have a deterministic equation with no stochasticity. And the deterministic equation would be called replicated equation. What we now do is also include the effect of the fact that the group sizes are actually not large. And if you do that, these are the equations we solved. Remember, there were 4 strategies, right? You can now, if you assume that the population size is fixed, I can reduce it to 3 strategies effectively. So we have this coupled SD of 3 strategies. First term captures selection, second term captures mutation, and then followed by drift. So the summary of this is that infinite population would not have sustained cooperation, but finite size promotes tag-based cooperation. And what is surprising is that the tag has to be costly. Tag is also called green beard in the cooperation literature. So what we found was that if there was no cost to be having a green beard, then cooperators are always outperformed by defectors. But the fact that there is a cost actually really helps them sustain cooperation. Okay. So, and that brings me to the really last part of the talk, which I will hopefully finish fast. Which again shows you in a much more general context. Now let's move away from the group in behavior. In a very general context, what are the ways in which the finite stochasticity can affect dynamics of populations? So let's consider this very simple dynamics of evolution, where birth, death, and mutation leads to changes in the frequencies of 2 alleles. Let P denote the proportion of type 1. And then this is the classic replicated equation that I just mentioned. So the proportion of the type basically is changes in a way that's proportional to differential fitness. If omega 1 is greater than omega 2, that guys will win. Now that's the sort of classic Darwinian prediction, right? So so basically in the previous figure I showed the 2 colored birds, right? So if the blue bird had higher fitness than the red bird, those guys would dominate the population over time. This is an example of a simulation. If you simulate this equation, you find this. Makes sense. Now, people have studied the idea of small populations for a really long time in evolution. That's called random genetic drift. So what does that mean? Even if the fitness are same, right, you can have evolution. And so usually the classic result is that the probability of fixation of any type is proportional to the frequency of the type. So for example, here I'm showing you 2 simulations where the 2 types have exactly same fitness for 100 individuals, and one of them has undergone a fixation. In another simulation, blue has undergone fixation. So it really entirely is random. Who gets fixated is entirely random, right? Something people know really, really well. But here is a very interesting catch. All of the theory of, not all, most of the theory of random genetic drift assumes that the population size is fixed. Imagine there are 2 competing types. There is a stochastic birth-to-death process. They have equal fitness. Okay. They assume that at every generation, the number of individual total population size is fixed. That's the assumption of classic Moran process and so on. Okay. And it turns out that that's not obvious. That's obviously not true. If you actually let two competing populations, even if they have equal fitness, when they compete, the total population size need not remain 100. It will go 95, 105. The population size can change over time is something I don't need to convince anybody, right? If you now explicitly account for this, some very surprising results come out. That's what I'm going to show. Okay. Here's the example. So this is an example where the blue type, this is an okay. This is an example where the blue and the red actually have same fitness. Okay, but you still find that the blue dominates over red every time. I mean, every time, a large majority of the time. Okay, in the sense that the probability of fixation is not equal anymore. So why does this happen? And in fact, this happened only for small population sizes. This doesn't happen for large populations. So in small populations, the prediction of the random genetic drift doesn't actually hold true if you allow n to fluctuate. So why does that happen? So to do this, again, we sort of followed our standard procedure. This is a paper which again came out just this year by an undergrad in our lab. And we called it noise-induced reversal of selection. So what do we mean by that? Okay. So here's the summary. Again, we write down stochastic birth and death process. We write down the master equation for the population densities. And then we use the system size expansion, write down the Fokker-Planck equation. And we then move from density equations to the frequency equation, which gives us the replicative equation. Okay. Let me just show you some interesting results now. Some definitions. So we first assume that there is a per capita birth and death rate we can define for each type. And this is a very general framework. We don't have to assume that just two types. You can assume arbitrary number of types. Okay. And we define two quantities. Fitness, which is the birth rate minus the death rate, and something called turnover rate, which is the summation of birth and death. So what we basically show is that it's not just fitness. The turnover rate also matters in random genetic drift. Let us see how that happens. So now, if you now follow all those steps, you find an equation of this type for population densities. Here is the first interesting thing. The mean expectation, the mean expected change does depend on the fitness difference. However, the variance depends on the turnover rate. Okay. Fitness controls the drift. Turnover controls the stochastic part. Okay. So how does that matter? How does that matter to ultimate outcome? Does it really matter? To do that, we have to now convert to population density to replicated equation. You use Ito's lemma. Okay, remember this is how the replicated equation looks like for the deterministic system. If omega is more than the average, you are good. You're going to get fixated. Okay. This is the frequency of I. If the frequency of a type I is greater than the average fitness, we'll have to go fixation. This is how the stochastic version looks like. Okay. Stochastic version has a drift term, diffusion term. Diffusion term here is not super important. But look at that. The first term here is basically same as this. The second, there's an additional term here, which depends on how the turnover rate of the type you're interested in depends on the average turnover rate in the population. So basically, ignoring the noise term here, what we basically find is that there are two ways. So the typical expectation of the replicated equation can change because of two ways. One is what we call an indirect mechanism, which is on an average 0, but not necessarily. So it's a bit subtle. I will not get into that. Today, focusing mostly on the direct mechanism. Let me just show you the example. What this basically, now let's consider this equation. Consider the scenario when two types have identical fitness. That means this term is 0. Even when two types have identical fitness, you can see that the rate of change of frequencies depends on an additional term, which depends on how the turnover rate is dependent on the average turnover rate in the system. Okay, and it has an opposite sign. So what this basically means is that the turnover rate is greater than the average turnover, its prevalence actually decreases. That means a slower turnover rate has an advantage over the faster turnover rate. So to sort of summarize this, there are two types, same fitness, but they have different turnover rates. The one with the slower turnover rate, that actually will persist in the population because it affects the drift dynamics directly. Okay.
Even if because in the turnover rate you have both the birth and the death, right?
Yes.
Even if the
but
birth rate is
that-
very large compared
It's
to the
but-
death rate, even if you're like.
So I'm directly comparing the turnover rates. So for example, you can take some examples like, take some examples where the omega 1 minus omega 2 is same for the, same. Omega 1 actually is equal to omega 2, which means that BI minus DI is same for two types. But if BI plus DI is different for two types, that seems to influence. The summation actually matters, not the difference alone. Okay, so this is an example. Assume n is finite and there is no selection. Let's say blue and red types have same fitness. Classic expectation is that the random genetic drift will now take over. Red and blue should have equal probability of fixation. Okay, but this is true only when n is fixed. The moment you assume n actually can change, we have the additional term here in the equation for the replicative equation. So this shows that if dp would actually be increasing at a rate that is non-zero when tau blue is different from tau red, which means summation of the birth and death rate of the turnover rate also really matters. And in fact, the one with the slower turnover rate is the one that actually is down. And that's precisely what happened here. So the blue had a slower turnover rate compared to red. And this effect is visible only in small population sizes. You can see this 1 over n factor here. So this you would not find in large populations. Okay. So sort of summarize the key predictions of the eco-evolutionary framework we developed is that the outcomes of larger and smaller populations need not be same if the turnover rates are not same. In smaller populations, smaller, slower paced life cycles will win over the larger paced life cycles, even if they have equal fitness. Okay. And in fact, a neutral evolution in such a case requires not only that the fitnesses are same, but also the turnover rates are same. Yes.
No, I'm sorry, I lost this because maybe you go a bit fast.
Yes, I'm sorry.
There's the one equation before you get to the stochastic equation for.
Which
Yes,
one?
This one?
This one?
No. After this one, no, before this one. Sorry. Yes, this one. In this one, right, you have only the fitness part, and then the noise is the square root of the turnover rate. But then you go to the next slide, and then the turnover rate is like go to the denominator instead of the numerator.
So it comes here.
Yes, this is why I a bit confused.
No, it is this is still in the numerator.
But what is the transformation here to get
So-
from that previous
so-
equation to this one?
So if I had a pen, I could show you.
Yeah.
So xi's are the
Is this just a Ito rule or what?
it actually is, yes. So xi is population density of n types.
Mhm.
P basically is a vector, which is P1, P2, Pn, where Pi is xi by summation xi. Right? So this is the transformation. I have SDE for xi's. I know it is SDE for Pi.
Okay. Thanks.
And remember that this is not constant.
Yeah.
So Ito's lemma acts very
Yeah.
strongly.
Mhm.
Okay.
Okay.
It's not constant. If this was a constant, I can just replace it by n and the SD for. And then of course, when I move from this to this, I have one more SD because technically there are now only n minus 1 terms in the proportions. I have another equation for dn. I have another SD for dn. Total population size. Yeah, yeah. Sorry, I lost part. I think everybody started answering with that. But I think the point is that this term is present only when we allow population size to also fluctuate, which of course is true in small population. And I think it dictates that the number of tigers when they are changing, they should always be at exactly 100, right? That's the main part. So I'll now show the summary slide. So sort of to synthesize all of this, I think the noise of finite system is really, really intriguing. Finite systems generate intrinsic noise. So we call this intrinsic noise because the stochasticity is not extrinsic, such as environmental perturbation, right? It's entirely because the birth, death, change of states, they're all part of the interactions of the individuals. And in such cases, strength of noise depends on system size and the state of the system, right? And that really makes it very interesting in the context of flocking systems. I mean, we think we have found three broad classes of flocking dynamics driven by stochastic finite size, noise-induced order, bistable, disorder and order, and finally deterministic order. And the sort of common feature across all of them is disordered state has more noise, and therefore you get pushed away from it. Although there is noise is not trying to push you in a specific direction, okay, it pushes away from disorder. Now, of course, whereas we can go in the state space, right? Now, and in the context of population dynamics, we find that finite size noise can also reverse the dynamics of selection. So once again, thank you all for your attention and your invitation. I really appreciate it.
Question for you. Yeah, can I ask you? So this is more like a general comment because it sort of hurt my physicist feelings in the beginning. No, it's because you said, the classical Turner-2 equations, you have this specific form of the potential plus these gradient terms. But actually, after Turner-2 put this out, people have written down the most general continuum macroscopic theory for polar order.
Yeah.
Okay. And that has a arbitrary potential and also has, even non-Gaussian noise. So it has it has multiplicative noise, but it has other corrections. So in fact, the most general continuum description allows for
all these possibilities.
All the things that.
Fair enough. Okay. I fully agree.
Yeah.
But one way I think about it is, of all those possible classes, what do real organisms exhibit?
No, sure. And I don't take away
Yeah.
that, but I was like, when you were framing,
I agree.
but then
I take your point. Yes.
yeah, but then my other question is, you only, your experiments, etc. It was very small number of individuals. I just wonder, are your equations scalable? Like, is the form of the drift term not having some other hidden term in there that when you increase n, you actually see it and might be exactly of the same form of the Turner-2?
That's a great question.
You didn't try putting more fish together.
I have two answers. So I'm almost certainly sure they do in many species. Okay. But it is also what I have shown is also descalable within some range. For example, I showed you this three graphs of this noise structure, right? Where the strength of noise were different for groups, different group sizes. So there we have done experiments with 15, 30, and 60 group sizes. Still small, but manageable in the lab, right? So up to that, they seem to work really well. And but as a very general rule, I would not certainly say that this equation will be true for all group sizes. I will not make the claim.
Okay.
I would not make the claim. It's entirely possible that at the larger group scales, maybe you will always get back to nothing. Possible. In fact, one of the experiments we are now trying to do is to do experiments with about 1,000 fish incredibly hard to do for logistical reasons, because it's very hard to get 1,000 fish of roughly same size at the same time. Because the aquarium fish is what we depend on. Aquarium fish market is what we depend on for these supplies. And why do you want 1,000 fish? Is what they want. Where do it's really hard for them to actually, and even if we get, many of them die. So it has been the challenge, but I think we would like to get there by. It is still not 1 million, right?
No, sure.
And several orders magnitude low, but better than. So it is something we are interested to understand. I would not claim that the same equation would always work.
No, but I understand. But just to follow up, so if you actually, I know you kind of ignore the spatial dependence of these things.
And that is precisely also what we want to measure from data. So we want to actually now do an experiment with thousands of fish.
Yeah,
I can divide the space into grids. Each local grid will have an auto parameter. I can try to measure the.
I understand, but it's at theoretical level. This is actually even though I know the Toner 2 quite well, I don't know exactly if people have figured this out, but at the theoretical level, if you have spatial dependence and you don't ignore the gradients, you can just study the dynamical stability of your system. Okay, so propagating modes, etc. And the form of that potential will have something to say about the stability of this system. And it could be, and this is the question I don't know, if I put a linear potential like the one you had, am I going to get some stable answer?
Yeah.
Because if I don't believe that is the right potential. I think I'm sorry, I'm being critical, but I wonder if people have studied the stability of Toner 2-like equations for different forms of that potential.
I'm not aware of the kind of study that you mentioned. Yeah, it would be interesting. I don't know.
This is something that Ramaswamy, if someone would have done, would be in that class.
Yeah. I would. Yeah.
You don't know. Okay.
I'm not familiar. Yeah, but but broadly speaking, I mean, there would be some fundamental limitations in taking this approach to the thermodynamic limit, because I cannot do such big experiments, right?
No,
I would be very limited. But from a
that,
purely theoretical point of view, I think it's interesting. I don't know about that.
Okay. Yeah,
that was
whether people looked at that.
Yeah.
Yeah, because I know people have been very much focused on the form of the Toner 2 potential and not explored other things, but maybe looks like there's a reason to do that.
Yeah, I can show you some other ideas.
Any other question? I just asked a small thing. Like in the end, they were talking about this slow turnover rate leading to fixation, right? But is there any intuition that we can think about, like why?
So maybe even I don't have a good intuition, but I think maybe what's happening is if there are two equally fit types, traits, so on long-term average, they are both subjected to some stochasticity. The faster turnover rate individuals would experience that very fast, right? So, and fixation is extinction is a one-way route. So because you are dying and giving birth very fast, although on an average that the same fitness as the other type, if I accidentally die, I can't come back. I think it is that reason why slower turnover rate, where you give birth and die slowly, would persist.
Because they experience noise
less frequently.
Okay.
Can I ask you? So do you have ideas for how you can realize the second part of your talk on the game theory settings in an experimental setting?
Yeah, it's very hard.
And related to that, I mean, is there a way you could see on trying to get the rule, not the interaction rule, but the rule by how they, are they trying to maximize? Are they really trying to maximize their profits? Can you, since you like to take stuff out of data. Can you take that that principle out of data? If you
Very,
want,
very challenging. Yeah, it's not easy though. Yeah, I would love to do. I've thought about it a lot. It's just not easy though. But what we can do is we can set up some ecological contexts such as foraging and predation, and then measure how well they respond to those conditions. That could be used as a proxy for their ultimate payoffs. That's the best I think one can do. But
so you mean out in the wild?
in the world, even in an experiment setup, for example, I give some foraging patch, right? And then we train fish in slightly different ways to mimic different strategies. And then you then let them compete. Whoever gets maximum foraging or fastest foraging. So then you can try to map strategies that you have trained to the efficiency of the performance. There are some, that is one way to do. But at the end of the day, I'm not a trained biologist, so I'm very scared of doing such experiments.
Is to train.
Oh yeah, very good. Yes, yes.
Yeah,
So
the
we
problem is that when
collaborate.
you put people in the lab or animals or whatever, and you say the goal is this,
Mhm.
then you turn on the goal. But you want to know, what, what, what is the goal that they're trying to make? Whether it's maximizing foraging in the long run, whether it's in a finite amount of time, or
Yeah, we don't know. Yeah. It's a very good question. Yeah.
Yeah.
So we have done that. The easy experiment to do relatively is to introduce some predation-like stimuli, which we have now data for 3 different systems. One is in collaboration with Guttal in France. There are the sheep that are chased by dogs. Okay. And then we try to look at how they are responding to the stimulus of a dog chasing them. We have done experiments with fish where we train fish to associate a stimulus such as some light, color light, green light, to an electric shock. And so every time there is a green light, you give a shock and train some individuals and do not train many others. And then when we do a mixed experiment with untrained and trained fish, provide the stimulus, the trained one would run away and then look at the response of others. And then try to measure infer some adaptive significance of social interactions in the process. And the third one, we have field experiments where I showed you, I don't know if I have the videos here, maybe no. I showed you these ungulates in the very early slide. We have black buck that are moving in the grasslands. We have drone footages of them. And then an experimenter walks towards them. That acts as a stress. And they all run away. Okay, now in all these different scenarios, from laboratory to field to very uncontrolled field experiments, we are trying to see how the information propagates and if there's some optimization that is happening. But it's really challenging to come up with such measures. But we are working on those.
Okay, so if there are no more questions, we can thank the speaker once again.